Thank you so much for having me here today. It's a real privilege to be able to zoom in and give you a bit of an update on research. As usual, I've been given far too much to talk about in not enough time. I've got my little timer here to keep me to the rule because I do tend to get gambling once I get going, a bit too enthusiastic. So my job today is to give you a bit of an overview of research from a global perspective, but also what's happening within Australia and within Queensland as well. Now, as I go through my talk, you'll see that I go from the international through to the more local aspect of research. But you'll notice as well that the lines are very blurred. I'm just going to smoothly transition from one to the other without you probably noticing, because that's the way research works. There is a huge amount of research going on within Queensland. There is an enormous amount of research going on around the world and around Australia, and they are working together. There are huge amounts of collaborations happening around the world, with Australians teaming up with international consortia of scientists to get the answers, because that's really the way research is going to move forward. And I think, as Lincoln mentioned, collaboration is one of the values of this organisation, but it's certainly a very, very important part of research as well. So, what are our goals at MSRA? They're preventing MS, better diagnosis and treatment, and ultimately repair and regeneration for the damage that's already been done. So ultimately we just want to kiss goodbye to MS, which is our fundraising awareness campaign that's taken on a national perspective. Now, in order to do that, we have to focus on different research streams, and MS is a very complex disorder. I'm sure you're all aware of that. And we need to tackle it from many different, from many different directions. We have to look at the immune system, we have to look at the brain, spinal cord, how nerves work, and how the support cells in the nervous system work. We need to look at viruses and other organisms that interact with the immune system. We have to think about new drugs, new chemicals that will help tackle those biological systems. And while we're doing all of that, we actually have to think about the people living with MS from a day-to-day perspective, because those four first dot points on the screen there are long-term pictures. We have to be patient and we have to look at the fine detail in order to get to the answers. And clearly that's not going to happen fast enough from the perspective of many of you. So there's also work to be done in terms of helping people to live better with MS, and that's what the social and applied research stream tackles. Now, as I mentioned, the work that we do involves neurobiology, the immune system, epidemiology, which is the study of environmental factors that impact on the body and contribute to disease. But, and all of those things are trying to answer our research questions. And I just wanted to put up this slide, which I shamelessly stole from Professor Trevor Kilpatrick in Melbourne, who is a very prominent MS researcher. And he wanted to look at it from the perspective of the questions that people with MS and their families ask. And you'll find that even though it looks like we're doing quite unusual and things in the lab that are very difficult to get your head around, they're looking at the fine detail of how the human body works, and it can seem very far removed from the day-to-day processes, the day-to-day experience of living with MS. But you'll find that those questions are the same questions that you're asking and that the researchers are asking. But we just have some rather fancy words to use to explain the different areas of research that we have to work on in order to answer these questions here. Are you having trouble reading it? Sorry, it hasn't come out very well. So basically at the top you've got etiology, the cause of the disease. You've got genetics, which is your DNA, how your body is put together. Epidemiology, which is the study of the environmental factors that impact on our body and contribute to our susceptibility to disease. Biomarkers, which enables us to track disease in real time. Can we find molecules in the blood or the cerebrospinal fluid? Can we use specialised types of MRI scans to visualise and see in real time what's happening? Biomarkers enable us to track whether a treatment's working or not. They allow us to maybe predict someone's course of disease. Will they have a mild course of disease? Will they have a rapid course? Will they develop progressive MS and so on? But we don't have those tools yet, but that's an area that is very important. Sorry, I'm trying to read them myself. Molecular pharmacology, so molecules in the body that interact with drugs, and whether you may respond or not respond to a drug is a very important area of research as well. Pathogenesis is the disease process in the body. What are the molecules that are working together to cause inflammation, to cause nerve cells to degenerate, to encourage new cells to regrow and regenerate the body, and so on. Another area is clinical trials, regenerative medicine, which I'm going to touch on again later on, and treating the symptoms of the disease. So those are all the fields, I'm sorry that you can't see it, that are written down there. And those are the areas that we're tackling to answer your questions, which are things like, why me? Why did I get it? Is my family at risk? How can you tell whether I'm going to have progressive MS or not? Is my therapy going to work? What can you do to repair the damage that's been done? All of those are questions I'm sure that you frequently ask, and those are the questions that we're working on with these different areas of research. And hopefully you'll see that one. So, on the international, global scale, particularly from the pharmaceutical industry perspective, there is a push, and it's quite amazing just in my short time working in MS research, that a number of new medications have already come off the end of the pipeline and into the clinic. And it's really quite exciting to see there's quite a long list of new treatments following on and about to come off the end of the pipeline as well. The first three listed there are very close. They're in regulatory authorities being assessed for introduction into the clinic in a number of different countries at the moment. 2 oral drugs there and another infusion, similar to the infusion of Tysabri that you probably know of. And then there's another few that are already in clinical trials, and we're awaiting the results of those trials. So I haven't got time here to go into detail about the different treatments that are coming through, but it's really exciting and really encouraging to see so many close to the end of the pipeline, and I think we're going to see some interesting changes over the next couple of years. So the list there that I just showed you, those drugs about to come off the end of the pipeline are all for relapsing remitting MS. And progressive MS is a bit more of a difficult problem to tackle when it comes to drugs and treatments. All of these drugs currently tackle the immune system, and progressive MS is more to do with the nervous system and the way that it degenerates and repairs. And that's going to take a different approach. Some of the drugs that are being investigated for relapsing remitting MS are also being looked at for progressive MS, and we're looking forward to the results of those trials fairly soon. But as I mentioned, progressive MS needs a different approach, and it's been a bit of a stumbling block, and it's been a serious frustration amongst the MS community that, why aren't there more trials for progressive MS? And it's actually now the subject of a major international collaboration that's been supported by the MS International Federation, which is a collaboration of MS societies from around the world. And they have really backed a group of scientists to get together and say, what are the barriers to conducting clinical trials in progressive MS? Why are they not happening? What can we do to get over those barriers? And listed here are the 5 main areas that need to be tackled in order to get over those barriers and get more trials happening for progressive MS. All treatments start in the laboratory, and at the moment for relapsing remitting MS there's a mouse model that has a version of MS. Some people argue that it's not really MS. It isn't MS, mice aren't human beings, but it has proven very useful in testing the current drugs that have come through, and very useful for teasing apart some of the immune system biology that is involved in MS. So that's one of the problems for progressive MS. There isn't a laboratory model that works very well for it. That's a target to overcome. Identifying the molecules that are involved in the degeneration and the repair mechanisms that are not working as well as they should is another hurdle. Clinical trial design for progressive MS is problematic. You have a much longer time frame to look at to examine progressive MS, and that can be very, very costly, which is a frustrating barrier to bring into it, but it is a big problem. And so finding the biomarkers, as I mentioned before, the systems that you can track in real time whether a treatment is halting progression or having an effect on progression is going to be a really key thing to being able to design and conduct clinical trials. The physical signs of progression to be able to be measured. And then for people with progressive MS, how can we better manage the condition during the course of their disease? So this is the focus of a big international collaboration, and I think we can expect to see some interesting developments coming from that in the next year or two. So it's really a case of watch this space. Now I just wanted to talk about the genetic discoveries. I'm sure you've all probably heard about this. It was later on in 2011, a scientific paper was published announcing that they had discovered 57 different genetic variations that contribute to MS. And this has been contributed to, this study involved many Australians with MS and New Zealanders with MS who donated their DNA to the research. The Australian researchers teamed up. They discovered several gene variations by themselves just by studying the DNA in Australia, but then they teamed up with an international MS genetics consortium, the IMSGC, and pulled all of their thousands and thousands of samples of DNA from people with MS and healthy control people, and looked for the genes that were more prominent in people with MS. And it's really an important milestone in MS research. It's identified that the majority of those genes are immune system genes. It doesn't shed any light yet on why some people get progressive MS and others don't. And that's an area that's going to require some more intricate genetic studies to do. And scientists in Australia and overseas are pursuing that as we speak. It confirms that why some of the drugs that will currently work, why they work, and it gives us clues about new directions that we can go in to design new drugs. Matthew Brown at University of Queensland Diamanta Institute was a major player in the ANS-Gene collaboration, the Australian-New Zealand MS genetic researchers, and a few of the other Queensland researchers are listed at the bottom there. We've got Lottie Tajuri, Alan Baxter, who's at James Cook University in Townsville, Simon Broadley, whose name I'm sure you know and I've spelled incorrectly, apologies, and Lynn Griffith at Griffith University. Now that research, it doesn't allow us to test a person and say whether they're going to get MS or not, and it doesn't allow us to identify family members that will get MS or not get MS. But it has enabled, just last month, Professor Broadley published a paper with colleagues from around Australia where they've taken 18 studies from around the world and combined the data from those studies, and they're now able to give us much more accurate predictions of family members, the risk to family members of developing MS. And they've shown with the data that the susceptibility to MS is accounted for, 50% of the susceptibility is accounted for by genes, and that the rest of it is due to environmental factors, which I'll come to later. And I won't go through the risks there, but you can see that for family members there is an increased risk in comparison to the general population. But we can now give a more accurate prediction of what the risk is, and hopefully when we have a better handle on interventions we can start thinking about how to prevent MS in family members. ANS-Gene is just one of the national collaborations that as part of my role to oversee, and there's a long list of them there that I don't have time to go into, but one that I did want to touch on is the neuromyelitis optica collaboration, which is again headed by Professor Broadley and his colleague Dr. Waji Bukhari at Griffith University, and they're working together with neurologists all around Australia to look at NMO. NMO is a demyelinating disorder similar to MS, but it's not the same as MS. Some of the symptoms and the diagnosis at the beginning can be very difficult to distinguish between MS. But it's very clearly linked to an antibody that attacks the cells in the brain, the support cells known as astrocytes. And this molecule that is being targeted by the immune system is called aquaporin 4. So what Simon and his colleagues are doing now is to try and characterise this disease. Oh, that's my time up already. Characterise the disease in Australia. find out how sensitive this antibody test is. Can we detect who's got NMO and who's got MS very early on in the piece so we can treat them correctly? Because NMO doesn't respond to MS drugs. So we need a different approach, and we need to identify those people early on. So this is quite an exciting area that, again, it's just starting out, and we've got a year or two to look for the results, get the results of this, this research. Judith Correa, you're perhaps all familiar with as well, at University of Queensland, and her PhD students here. They've been looking at, again, at antibodies that attack the body's own molecules. These pretty looking cells over here, this is an oligodendrocyte that makes myelin. This is an astrocyte that supports the brain cells. And in the lab we actually use antibodies to label cells and make them show up under the microscope. But Judith is using this very same technique of staining cells with antibodies to find out what self-directed antibodies there are in the blood and cerebrospinal fluid of people with MS. And that will give us clues about how the disease works, what's happening in the immune system. Michael Pender at University of Queensland is pursuing his research into EBV, which is the virus that causes glandular fever in young adults, so-called kissing disease. In children, it just causes a sort of non-specific viral infection. About 95% of the population will have been exposed to EBV virus in their lives, but not 95% of us get MS. So we need to work out why some people who are exposed to EBV develop MS and why some don't. Michael has got some evidence to show that the immune system in people with MS is not so good at clearing this virus when it's hiding inside B cells, part of the immune system. It's hiding in those cells, and the T cells are not so good at clearing those infected cells away in people with MS as they are in the general population. And Michael's just pursuing that research still, and he's getting some very interesting results. And this links in very nicely to some research that's going on in the UK and other parts of the world, looking at potentially developing a vaccine to EBV and preventing MS, and also whether antiviral treatments might have any role to play in treating MS. This is all very experimental, very early stages, but it's going in a very interesting direction. Jun Yan and Jason McKenzie in Queensland are studying the sort of the functional end of the genetic story. We know what genetic variations make us susceptible to MS, but we don't know how those genes work. What are they doing inside the body that makes you more susceptible? So Jun and Jason have been investigating that end of the scale. Again, making some interesting progress and helping us to understand how the immune system works in MS. So, as I mentioned, with genetics area of MS research, we've got the genes, we know what most of the genes are now, we need to understand how they work, what they do in the body, and that's a major focus for many scientists, particularly in Queensland and around Australia. The National Health and Medical Research Council just announced their funding just last week, and they have given $3.5 million to MS research just beginning in 2013, which is really exciting news, and that's just a little bit of an increase year on year over the last few years of money going into MS research, which is really encouraging. Many of those researchers receiving that funding have previously had MSRA grants, and with the help of MS Queensland and the other MS societies, we've been able to fund that research and build capacity for MS research in Australia, so that's really exciting to see those results. I really should shut up, but I just want to tell you a little bit about the next areas of research, which is repair and regeneration is obviously the kind of the ultimate goal. The drugs we have at the moment are very good at stopping the inflammation and suppressing what's happening with relapsing and remitting MS, but we really need to be able to repair the damage that's already been done for so many people. Stem cells are going to play a major role in this area of research, either through getting the stem cells in your own body to be better at repairing. We have the capacity for repair in our bodies. Sometimes the environment doesn't allow those cells to grow and regenerate, and we need to learn how to encourage them to do that. And sometimes they need to get to the place where they need to go. And so there's a lot of research. Melbourne is particularly strong in this area of research at trying to get the body's own immune stem cells to do more repair, do it better. And there's also a focus on transplanted stem cells, either bone marrow stem cells or mesenchymal stem cells, which come from many tissues in the body and have particular characteristics. Animal studies and some very early clinical trials have suggested that stem cells that are transplanted into the body don't actually take on a new role. They don't grow into new nerve cells or new myelin-producing cells, but they help to make an environment that's calmer from an inflammation perspective and more permissive, more encouraging for regrowth of the cells that are already there. So that will be an interesting area to watch. And neuroprotection is really important. There's a cascade, and once a nerve cell has been stripped of its myelin and damaged, there's a cascade of molecules that sort of dismantle, if you like, the nerve and remove it. And we need to stop that process. And Stephen Petratos earlier this year discovered a molecule that can do just that. It works beautifully in mice. Again, they're not humans, so we have to watch this space. But the drug that he used to block degeneration in mice is actually already approved for another nervous condition. Alzheimer's disease is one of them, but other neurodegenerative conditions. So it's still got a lot of preclinical work to do before clinical trials of this molecule can happen in people with MS, but it speeds the process up because that drug's already jumped through some of those regulatory hoops. Stem cell transplants I touched on. Claude Bernard in Monash University is doing some excellent work on that, and he's collaborating with researchers at the Californian Institute for Regenerative Medicine, which is the biggest centre for regenerative medicine in the world, and he's working with them on working towards clinical trials for stem cell therapies. I've mentioned social and applied research and trying to understand the impact it has. A lot of the research that we have been doing with the Australian MS longitudinal study has generated the data and the evidence that we need to be able to support you and advocate for you with government and improve services and getting access to those things that will make life more straightforward and easier for everybody. And some of those areas are listed there, and there's some areas of clinical research that allied health, physiotherapy and support of people with MS. Those areas of research are continuing. I just wanted to finish with the biggest and the most exciting thing that I'm working on this year, together with researchers and clinicians from all around Australia, and this is our prevention trial. The body of evidence now about vitamin D in MS is huge. We need to find out if vitamin D can do anything to slow down or prevent the development of MS. International researchers have been struggling with this one for many years. Getting the funding to get a clinical trial like this off the ground has been a huge challenge, but with the help of MS Queensland, MSWA, MS Tasmania and some other trusts and foundations, we've actually got the funds now. The researchers are already in the process of setting this trial up, and we hope to enrol the first patients in December. Thank you. This is not for people already diagnosed with MS, but people who have had their very first episode of symptoms that suggest they may develop MS. And we will give people vitamin D of 3 different doses and a placebo, because that's the gold standard randomised controlled, placebo-controlled trial to investigate whether vitamin D can prevent a second episode or delay it. And we need to know this. Many people with MS are taking vitamin D, taking all sorts of different doses of vitamin D. We don't know if it's safe, we don't know if it's effective, and we really need to know what benefits we can expect. So we're going ahead with this trial, and we will have to wait a few more years to get the results, but it's something really exciting, something I'm very proud to be involved with.